Hello, everyone. I'm Alain Chelotal, and I will be hosting the new Worldwide Neuro event today. So we're happy to have Sten Linnarsson from the Karolinska Institute in Stockholm. So Sten did, for those of you who don't know him, he did a PhD with Patrick Ernfors at the Karolinska Institute, working on neurotrophins. And then, so I had the confirmation. He didn't do a postdoc. It was quite unusual. He started a company, so which is an interesting way of doing science. and then he escaped somehow, and he went back to academia, to again back to the Karolinska, where he studied first, and now he's professor there. So clearly, I mean, Sten, I mean, I've known him a little bit now. He's one of the, I would call them the Swedish wizards, or the, I don't know, the Vikings of RNA-seq. So all this wave of people from Sweden who have really made a huge progress in the way we do biology now. So they're really the pioneer in terms of technique. And in the case of Sten, it's mostly on single cell technologies and in particular the application to neuroscience. So, I mean, if you look at what he has published, it's really impressive. The papers, I mean, I'm not a big fan of citation index, but I mean, all his papers are really like key papers in our field and many other fields. So he's really highly cited. And with the work they're doing there is really changing the way we see the brain. I mean, where I started, and many of you, there are a few cell types, a few astrocytes, a few oligodendrocytes defined by morphology, many neurons, but not so many types. And now with the work he has been doing with his colleagues, I mean, everything is exploded. We have many, many new cell types, and it's really a very interesting time. And as you will see, it's even more diverse during development. And what I really like about these papers, and you will have some examples. I mean, I don't do much single cell now, and it's often not boring, but the way the data are presented with t-SNE and UMAP, it's not so appealing for an anatomist, but he always have some nice figures in his papers, and you will see an example with a planisphere, I guess he will show it, that are really like almost artistic. So it's a very nice way of showing the beauty of that type of analysis. So just a few things I want to mention. So he's also leading a big EU consortium that is named Brain Time, and I think he will have, maybe he will present you what they're doing. And also he has been one of the founder and one of the pillar so far of the Human Cell Atlas, which is also a very interesting initiative. So Sten, I'm going to give you the stage, and it's the first time I'm sharing, so I hope it won't be a big mess. Okay, thank you, Sten, again.
Thank you. Thanks, Alain, for the introduction, and thanks for inviting me to this forum. I hope you can hear me, and I'm going to start sharing my screen. And let's see if it works.
Yeah, perfect.
I hope you can see Big Brain. Yes, good. All right, so the theme for today is going to be brain development in space and time. I'm going to talk about a project that we are just finishing, that's been going on for several years, of trying to map brain development using single cell technologies. As Alain mentioned, I've been working on mapping various parts of the brain and also doing brain-wide surveys of characterizing cell types and sort of trying to understand the system behind it all, developing taxonomies of cell types and so on. But when you go to development, you quickly realize that you cannot really compartmentalize cells into distinct types. Everything is in motion, everything is distributed in space, cells are maturing over time. And so it's actually several dimensions in addition to what you would find in the adult brain. I find it extremely interesting and stimulating, also incredibly difficult to work with and to visualize and present anyway. So I'm going to try to do my best and see how it goes. All right, so for today's theme, we're going to focus on space and time, because I think they are probably the most important aspects of how the brain builds itself. Of course, in time, there are sort of waves of key events in brain development. There is the initial formation of the nervous system, neurulation, followed by a long wave of neurogenesis, building all the various kinds of neurons that we need in the brain, which are then connected, and they migrate into the right places and so on. And towards the end of gestation in mice, the progenitors switch to start to make glia in the gliogenic phase, and oligodendrocytes of course then start myelinating neurons and so on. So it's this very exquisite orchestrated sequence of waves of building the brain. And then on top of it, you have non-neural cell types like the vasculature and the immune cells that at the same time have to invade the brain and sort of populate it. The same general scheme is true in humans, although the timing is vastly longer. Everything takes longer, but it's kind of proportional. The other aspect is space, and it's one of the most important aspects of brain development, of course. The most canonical example is the dorsoventral patterning of the neural tube, which is, I think, best studied or most well studied in the spinal cord, where morphogens are secreted dorsally and ventrally and generate these opposing gradients, which are then read out by gene regulatory networks that sort of lock into specific combinations of transcription factors at different distances from the dorsal or the ventral side. So you get these beautiful sort of layers of cells or progenitors that are then poised to generate different types of neurons. But this is, of course, just one place in the brain, and it's just one axis. At the same time, similar patterning events are ongoing everywhere in the brain, and they're not just dorsoventral; they're anterior-posterior, and they're much more complicated than that. When the brain acquires its a complicated three-dimensional shape. So we're going to try to take one step towards understanding this on a brain-wide level, but it's clearly an enormous challenge. Right, so I'm going to focus today on a single-cell atlas of the mouse brain development. An initial version of this is out in bioRxiv, but we have many updates, so I hope that even if you've seen this before, there's going to be several new things here today. This work was led by Joelle Lamanno and Kibli Seletti in my lab. Joelle is now a PI at EPL. And also with Alessandro Furlan, who from the Patrick Anforth group, and many co-authors. I will try to acknowledge everybody as we go along. So in this project, we sampled the mouse brain from embryonic day 7 until birth. So we only have pictures of up to E16 here, but we actually have up to E18 we've sampled. So we sampled every day, 7, 8, 9, 10, 11, and so on. Initially, we sampled the whole embryo, and then the whole head, and then the forebrain, midbrain, hindbrain separately, and so on. So we sort of refined the dissections as development progressed. So you can see here in this summary how, for example, at E7, we have 3 independent samples. Each of them is a whole embryo, whereas E8, we have 3 independent samples. Each of them is a whole head. And so on and so forth. As the brain grows, of course, we sample more embryos to have greater coverage and sort of match the complexity of the nervous system as best we can. In total, we sampled about, I think it's 75 samples. Some of them are multiple embryos. And we sampled about 300,000 cells. So if we start with an overview of the whole data set, a Disney plot of all the cells that we have in the data set. It's, as I said, 300,000 cells approximately, and laid out here and colored by a major class of each cell. So what we've done here is we've gone through the data, we've clustered it, we've done QC, removed doublets, removed bad cells, then we've clustered it, And we've made a big table of all the clusters that we found. We find nearly 800 different clusters. And for each of them, we've used prior knowledge and literature and marker genes and so on to define the major class of each cell. And so this is what we came up with. And I'm going to give you a quick tour of this map here, and we're going to refer to it during the talk. First of all, sort of the central component here represents the early embryo and the neural tube and everything that derives from it. And then you have these islands around, for example, here, these are blood cells, vascular cells here, you have immune cells here. And so these are cells that don't derive from the neural tube and therefore they're disconnected. We have a major component over here of neurons, which are cells that already express some markers of mature neurons, although, of course, there is long way to go for these neurons to become really adult, mature neurons and to be fully connected and so on. At the other extreme, we have the early embryo down here in gray. We call it gastrulation now, but I'll go into a bit more detail about what we can find here. A different way of visualizing the same data is to show the change in proportions of all the major cell classes over time. So now I'm showing the proportions of all these classes for each day from E7 to E18. And you can really start to see these waves that I talked about in the beginning that coordinate and sort of unfold brain development. So it starts with these early cell types that we put now under the general heading of gastrulation. And quickly at E8, we see formation of the neural tube and of some other tissues like ectoderm, so not neural ectoderm, but other ectoderm of mesenchyme and so on. And then just one day later, you see the emergence of radial glia and this big wave of radial glia that will make up the bulk of the neural tube for many days, for about a week of development here. And these are really the cells that self-organize, induce each other to distinct fates and will generate a large proportion of the diversity of the brain. Radial glia then eventually differentiate into neuroblasts. So you have this wave of neuroblasts, and neuroblasts then become neurons, and at the end we end up with mostly neurons, although there is still some ways to go here for development. Also towards the end of development, towards birth here, you see the emergence of a subtype of radial glia that we call glioblasts. So these are the radial glia that are now likely to have stopped making neurons and started to make astrocytes and oligodendrocytes. We don't really see many astrocytes here, not enough to plot on this diagram. We do see a small sliver of oligodendrocyte lineage, so these are mostly oligodendrocyte progenitor cells, and yeah, some other minor cell types. Now, putting this all together, we tried to make a visualization of brain development that will combine time and differentiation and both the sort of continuous aspects of development, but also the discrete classification into classes and cell types and so on. So inspired by old medieval maps of the Earth and of the planets, we made this visualization that now I'm going to walk you through to describe what we're seeing. So in the middle, we have the Disney plot. It's the same as I showed before. Now I've labeled it on top instead, and I've added a bit more detail. So again, you have the early embryo down here, which is divided into germ layers, and you can see structures like the node here, for example. As development progresses, this is replaced by the neural tube, ectoderm, mesenchyme, neural crest. So these are sort of early structures of the head of the embryo. And there is a first branch here where a fibroblast lineage branches off from the neural crest and the mesenchyme, which will eventually develop into the pia and the arachnoid, so the meninges. But the main trunk of the development process here is the maturation of the neural tube into radial glia that will generate all of these neurons. So, for example, this big branch here is forebrain excitatory neurons of the cortex and hippocampus. And this big cloud is everything else. So, for example, here you have the cerebellum, here you have the forebrain GABAergic neurons. So this is sort of the bulk of the brain generated here. Towards the end of development or of prenatal development, radial glia differentiate or mature, I should say, into glioblasts, which will go on to make astrocytes and OPCs and some ependymal cells. Now, this is 300,000 cells here. We clustered them, as I said. We obtained almost 800 clusters, and they are shown in the dendrogram around the circular dendrogram here. So each leaf of this dendrogram is one cluster, and we've cut it into 25 clades or 25 metaclusters here for clarity, and I'll take a moment to go through to explain what we're seeing here. So the dendrogram starts here, and it goes all the way around and ends here. The first two clades here are cells of the gastrulation stage. So this is, again, the early embryo here. This color here corresponds to the class on the main Tisney, so you can follow along. There's another clade here of still early embryo, but a little bit later, where you have mesenchyme and neural crest and ectoderm and so on. Then come the blood cells. So these are erythrocytes. Actually, they form two distinct clusters. You can see also here, two distinct islands.
Sorry, Sten, we don't see the pointer. I don't know if you...
Yeah, all right. That's too bad. I don't know how to make it show up.
So it's okay, but just keep...
Yeah, right. Then I have to explain instead.
A bit more, say, yeah, the quadrant. For now it was okay, but here it's a bit more complex. But go ahead.
Okay, yeah. Yeah, right. So for each clade, and now we're looking on the bottom left there at the blood clade. You can see the colored dots on the clade on the outer perimeter, which are red in that case. They correspond to the colors in the Tisney in the middle. And for blood in particular, we get two major distinct clusters, which correspond to primitive and definitive erythropoiesis. So the way that development works is that there's a first wave of primitive erythrocytes that are nucleated, and this is replaced quite abruptly, actually around, I think, E13, E14, by definitive erythrogenesis, which works the same way it works in the adult. Then, if we follow along the circle there, the next step is the fibroblasts. This corresponds to what I mentioned before, the fibroblast lineage on the left of the Tisney there, which will mature into the meninges. Then we have vascular cells and immune cells, which of course originate from outside the nervous system. Then we come to the radial glia. You see three major clades there. It's some 50 different clusters or something like that. And they correspond to, again, to the trunk of the Tisney in the middle, followed by a smaller clade of glioblasts. You see the OPCs there. There are Schwann cells and olfactory sheathing cells. On the Tisney, they locate quite close to the glioblasts, but there is a gap in between, and we don't think they're connected. They are actually supposed to be neural crest derived. It's just that in the way we sampled the tissue here, we don't probably have the intermediate cell types there. Then on top of the dendrogram, you have a large chunk, maybe 30 degrees of the circle here of neuroblasts in violet, followed by the neurons, which are the most numerous clusters, and it's almost 1/3 of all clusters here in this data set that are neurons. In the four corners, there is annotation. So, for example, lower left is the age of each cell. So you can, again, see how time sort of progresses from the gray embryo at the bottom and then branches out into the meninges, the glia, and the neuronal lineages. Top left, you see the region annotation. So since we dissected regionally, we know the origin of each cell. And for example, you can see the forebrain excitatory neuron branch there, the big branch on the top right, is purely from the forebrain. Bottom right, you see cell cycle. This is a cell cycle score that's based on the expression of cell cycle genes. And you can see that in all lineages, meninges, glia, neurons, differentiation happens in synchrony with the exit from the cell cycle to a large extent. All right, so let's talk about some general properties now that we can glean from this experiment. So first interesting observation here is that the total RNA content of a cell changes over developmental time, and quite a lot actually. So on the left-hand side here, you see histograms of RNA content, and we use the number of detected RNA molecules as a proxy for the total number of RNA molecules. Of course, we have losses and so on, but since everything here was done uniformly, we expect the losses to be also the same for all cells. So we think that at least the relative proportions here will be comparable. So for instance, at E7, you have most cells have a relatively large amount of RNA molecules in it. Whereas if you compare with the late time points, E15, 16, 17, 18, most of the cells actually now have less than half of the content of one of the earlier cells. Maybe it's easy to see on the right-hand side that shows the number of UMIs. So these are unique molecular identifiers. That's basically a proxy for number of RNA molecules in these experiments. And you can see in the early embryo, in the vascular cells, and in early radial glia, the counts are high, and then they drop both in the meninges, in the glia, and in the neuronal lineages there on the right. And the same for the total number of detected genes. So we detect many more genes in the early cells, in the progenitors, than we do in the neuroblasts and immature neurons. Now, what drives this difference? So one thing that correlates with it very strongly is cell cycle. As I said, we can calculate the cell cycle score on the left here by basically measuring the proportion of all reads or all UMIs that come from cell cycle genes. And you can see that if you make a histogram of this number, so you take each cell has, let's say a cell has 1,000 detected molecules, and let's say 100 of them are from cell cycle genes, then the score here will be 10%. Right, so if you do this kind of scoring for all cells and you make a histogram, you see that it's strongly bimodal, and so on the left you have cells that have very little cell cycle gene expression, and on the right in yellow you have cells that have between 1% and 2% of cell cycle gene expression. If you map this histogram now onto the t-SNE, you can see that the yellow high cell cycle gene expressing cells are from the early embryo and from the radial glia. And then as the cells differentiate in all lineages to the fibroblasts on the left, to the glia on top, and to neurons on the right, they exit the cell cycle at about that stage. Another way to see it is to split the histogram by age, as I've done on the right. So you can see, for example, the darkest curve here, the black curve, is E7, where almost all of the cells have between 1% and 2% of cell cycle genes. And that's true also for E8 and E9 and to some extent E10. Whereas if you go to E13, 14, 15, 16, almost all genes have less than 0.5% of cell cycle genes. So there is this abrupt switch in the whole brain in all lineages where cells leave the cell cycle. Another kind of surprising observation related to this, because of the way we do the sequencing here, we get reads mapping not just to exons, but also to introns. So we can measure the proportion of, for each gene, we can measure the proportion of reads that are from nascent RNA, so unspliced RNA, that is still likely in the nucleus and not yet spliced, and so it was just transcribed, versus spliced RNA, which is likely in the cytoplasm and has been around for a while, and so it's a bit older. And on the left, we plot this proportion, and you can see that in the early embryo and in the radial glia, this proportion is low. whereas in all the other lineages, as the cells mature, in the fibroblasts, in the glia, and in the neurons, the proportion of unspliced RNA increases, which I found kind of surprising. And interestingly, this correlates very well with the expression of spliceosome genes, which I show on the right. So when in progenitors that are still proliferating, the spliceosome is expressed at a high level, relatively speaking, and then it's downregulated. And so it makes sense then, or a possible mechanism would be that splicing rates are high in progenitors, so they express high levels of RNA, but this RNA is spliced rapidly so that there is a lower proportion of immature RNA in those cells. So they seem to have, they have higher total level of RNAs, they are highly cycling, and the genes are spliced rapidly. So it's kind of a very active state that switches off in all lineages as they differentiate. All right, now let's get into the biology of this tissue, and let's start with gastrulation. I'm going to go quite quickly over gastrulation, but these are sort of, we call this the gastrulation phase, but of course it's a bit more than that, and it's a bit longer, but it's the earliest time points that we have in this data set. It's from E7 and E8, and it corresponds to what you see in B there, the earliest stages of our data set. We are lucky that there's been a paper from Bertie Götkens and Marioni lab where they studied gastrulation specifically, slightly earlier time point than we have here, so E6, E7 approximately, and so we could map our data to their data, and indeed it matches very nicely. So in D and E, in D you can see their data set, and in E you can see our cells of E7 and E8 embryos mapped onto their plot there. You can see we have representation for all the lineages. We're missing the blood progenitor simply because we didn't include them in the analysis here. Erythrocytes are difficult to work with because they express so few genes and at extreme high levels, mostly hemoglobin. In F, you can see our clustering on the horizontal direction, and the clustering from this previous paper in the vertical direction. So you can see that the correlation between our clusters and their clusters is very good, and by and large we have observed the same clusters. We have a bit different resolution in different parts of the data, but otherwise it's very reproducible. Focusing now on, at the same time point, only the neuroepithelium. So now I'm taking out only the neuroepithelial cells. So we're very early in the formation of the neural tube. We observed these clusters that are shown in G here on the top left. And we wanted to ask what drives this cluster, what's the difference here that we observe? And by and large, the difference is driven by, as you can see in H, transcription factors mostly, and other genes that are related to spatial patterning. So for example, the first cluster there on the top expresses 6-3, which is a forebrain or anterior transcription factor. Whereas, for example, the third from the bottom, NEPID-C, expresses Hox gene, so it's a hindbrain or spinal cord, or at least a caudal cluster. So we think that we're seeing here, and you can see also in the Tisney plot in G, that it's kind of organized with anterior on the right and posterior on the left. All of these are very immature neuroepithelial stem cells or progenitors, but we wanted to ask if there would be already at this stage some cells that we would have called neuroblasts at the later stage. So cells that have already exited the cell cycle, already started to differentiate towards neurons. And so we searched for those cells that were part of clusters of neuroblasts that we observe only much later, or only later at least, but where there might be a few cells in those clusters that are aged E7 or E8. And indeed, we found a small number of cells, which are shown in J here, that are already neuroblasts and they're part of neuroblast clusters, but they are, as I said, from E7, E8. And we found cells from 5 different clusters, and interestingly, 2 of them are from cranial ganglia, so likely to make sensory neurons of the cranial ganglia. And generally, they're all from, as you can see in K, almost all from midbrain and hindbrain cell types that is in agreement with the idea that the midbrain and hindbrain develop sort of faster than the forebrain. A couple of short stories about lineages that are interesting from brain development point of view. The first one is the fibroblasts and the meninges. So it's thought that the meninges consists of 3 layers: the pia, the arachnoid, and the dura. They consist of fibroblast-like cells that form these membranes, and it's thought that they develop from the neural crest and the mesenchyme of the brain. And so here we took out this, as you can see on the left, this sort of teal-colored part of the tisne that was classified as early fibroblast and as pia and arachnoid, and we did a subclustering, and this allowed us to identify both early fibroblasts and sort of more mature and differentiated fibroblasts. We identified both the pia, the arachnoid, and the dura, and also a cluster of perivascular fibroblast-like cells, which are probably likely to be cells that sit along vessels in the brain. So they're not really part of the meninges, but they sit sort of along the vessels that penetrate into the brain itself. And we were able to identify then markers of both sort of the putative progenitors, the early fibroblasts, for example, MKX, and of the arachnoid and the pia, the 2 layers that we have most highly represented here. Collagen 15 for pia and the solute carrier 22A2 for the arachnoid. So our proposal is that we're observing a lineage here. We aren't able to confidently place the dura here. It's kind of disconnected. But for the pia and the arachnoid, it seems that we have a progression from a common progenitor that branches out into these 2 layers. So we wanted to validate that. And so Elin and Alejandro in my group performed the multiplex single molecule FISH using markers of both this sort of putative common progenitor and of the 2 layers. And I think what you can see if you focus, for example, on the first row here in G, you look at MKX, this is supposed to label the progenitors, the shared progenitors. And it can be a bit hard to see here, but what you're observing there is on the left is the head, on the right hand is the face. So the head is sort of looking to the right. There's a cartoon on the right that's on the far right that you can use to orient yourself. And indeed, we observe MKX staining in the future head region and in sort of the surface of the future head region. And the other genes that also label the same population are in agreement with this. If we now go all the way to the end, to make a long story short, and you look at collagen 15 and solute carrier 22A2, so those are the green and the red ones, and also at the merge in the bottom right, you can see that indeed at that stage now we are staining nicely separately the pia and the arachnoid. So while, of course, we didn't do lineage tracing here, we cannot prove that these cells are a physical lineage. At least these stainings are in agreement with that hypothesis. We saw a similar thing, actually, interestingly, with the immune cells. So in the brain, of course, you have mostly microglia and macrophages. And indeed, we observe mostly microglia and macrophages. The tisne plot in A here shows 2 large clouds. On the left, you have microglia and the smaller subtype called axon tract associated microglia, ATM. And on the right, you have perivascular macrophages. But interestingly, we observe a kind of intermediate cell type that we call early macrophage here. And if you look on the far right, you can see that type is the earliest. It emerges already at E8, E9. And we have a very suggestive branching lineage here that suggests that these progenitors, a shared macrophage progenitor, will differentiate around actually quite late around E14, 15, 16 into microglia and macrophages. There's an intermediate cycling stage there. We do observe some other immune cell types, but in very small numbers. We have some T cells and platelets and other things. All right, so that's the early stages of brain development and some of the non-neural lineages. But let's now go back and focus on this issue about space and time and patterning and so on. So we wanted to look now at the stage where the brain is patterned and where we believe that all the sort of major lineages of neurons are being specified and laid down. And the textbook description is that there are these organizers, so there are these cells that are progenitors that sit at borders and limits, as I mentioned in the beginning there with the spinal cord, for example. They sit at the limits of a tissue and they secrete morphogens that diffuse and induce different fates at different distances. So we wanted to sort of systematically search for these. Of course, there isn't a general definition for what an organizer is. It's kind of a functional definition. But the known organizers, they secrete well-known families of morphogens like FGFs and BMPs and WNTs and hedgehogs and so on. So we ranked all the radial glia cell types around this age, E9, E11, by the expression of these different morphogens. And we were able to then identify clusters, as you can see in A, clusters that score highly on the expression of these morphogens. And in many cases, then the red ones, we were able to identify these with textbook known organizers by the use of other markers. So looking at these now, we have a rich repertoire of putative organizers, and we can classify them roughly in ventralizing and dorsalizing. So ventralizing in yellow, so now I'm in panel C here on the right. Ventralizing in yellow are the ones that express sonic hedgehog, which tends to induce a ventral fate. And dorsalizing that express BMPs, for example, and some WNTs. There are these boundary organizers, the isthmus between the midbrain and hindbrain, or the anteromedial cerebral pole at the very front of the telencephalon in blue, which tend to express a variety of FGFs. And then at a later time point, the antihem of the telencephalon, which expresses neuregulins. So what's interesting about this is I think that we have a much richer definition or richer repertoire of organizers than you would find in textbooks, and we have for each of them a large number. We identified we defined 119 genes that were transcription factors or secreted molecules or ligands that are specific for one or another of these putative organizers. But it also now lets us start to understand sort of how in space is this organized, this rich repertoire of organizers. And to achieve this, we teamed up with Mats Nilsson's group, with Daniel Gyllborg, Kristoffer Mattsson, Langset, and with Elin from my group, to perform an in situ sequencing experiment. So this is the method called HUB-ISS, in situ sequencing. We performed in situ sequencing on a whole mouse embryo at E10.5. We scanned a total of 24 sagittal sections, which you can see down top right. And they're located about 40 microns apart and going from more or less the extreme side of the embryo and all the way over to the other side of the embryo. And we did this for these 190 genes that we defined as enriched in one or another of the putative organizers. On the bottom left is an example of the kind of data that we get from this. showing 5 of these 119 genes, and I think you can appreciate the enormous complexity and spatial precision that these genes show. And based on gene expression patterns, we manually curated an anatomical annotation using the Allen Brain Atlas nomenclature and ontology that we overlaid onto the section, so we can orient ourselves and know where we are. So that's what's shown on the bottom right there. So as one example here of a complete stack of images, on the left is the anatomy superimposed on the complete stack of 24 sections. And there you have telencephalon in brown and yellow, in brown, diencephalon yellow, midbrain in green, and hindbrain in violet. On the right is example of one gene, sonic hedgehog. So this is the canonical ventralizing morphogen, and it's expressed, as you can see, in the ventral part of both the forebrain, midbrain, and hindbrain. On the right there, I'm zooming in 2 steps, so you can see the individual RNA molecules that are detected, so you can see kind of the resolution that we have. It's also expressed in the notochord. So down there along the spinal cord, you can see a thin, very intense, thin line of staining. That's the notochord. It's also expressed in the ventral part of the spinal cord, of course. And this staining corresponds very well with the staining in Allen Brain Atlas, as you can see there on the bottom right. Another way to appreciate the richness of this data set, here I'm showing, again, a small set of genes. I'm sorry, the legend seems to have been lost here. But anyway, I think you can still appreciate the rich data that you can achieve in this way that would allow you to now place individual cell types or clusters by using the combinations of genes that are expressed in any given position here. So that's the next task that we set ourselves, is to now try to place all of the clusters that were defined before, not just have them be abstract clusters on the but really place them in space on this full embryo. So we tried a couple of different methods, and the one that we really came to like a lot is Tangram. So Tangram is a method based on gradient descent. So it's a model that tries to optimize the allocation of single cells to spatial voxels in such a way as to both make the density uniform. So the cells should sort of cover the whole space. You should not put all the cells in the same place, but they should cover the embryo. And at the same time, the gene expression vector of the cells that are in any given place should match the gene expression from the spatial data in that position. So this is there's a bioarchive paper. It's from Aviv Regev's lab, and it's I understand work led by Tommaso Biancalini and Gabriele Scaglia. Now there is a problem here, which is that Tangram computes a projection matrix that you can use to project cells to voxels. So this is a matrix that has on the right-hand side here, the size of this matrix is number of cells times number of voxels. Our problem is that we have 300,000 cells and we have 750,000 voxels after making a 30 micron grid on this data set. So that's a 900 gigabyte matrix, and I think it would take a very, very long time to optimize. And I actually didn't even try because I don't have 900 gigabytes of RAM. So the solution then is to map clusters instead of cells. So then you have 800 clusters instead of 300,000 cells, and the whole thing can be done in 10 minutes on a laptop. This requires one change to the Tangram algorithm, which we've implemented, which is that implicitly Tangram, as I said, it wants to allocate single cells to voxels, and it kind of wants to place them uniformly. If you now use clusters instead, you have to take into account that some clusters are large and some are small. So a large cluster, a cluster that consists of many individual cells, should automatically take up more space in space. Otherwise, you would sort of overpopulate your space with the tiny cluster. So we generalized the Tangram algorithm to take a volume prior, so you give it the expected size of each cluster, and then it really works beautifully for this purpose. So here's an example of the spatial probability distribution of one cluster. So this is what Tangram produces. It produces a map over all the voxels of the probability that the cluster is located there. And so here on the left is an example of a cluster number 206 that we had annotated as radial glia of the midbrain and diencephalon basal plate. And indeed, it's precisely exactly located to the midbrain and diencephalon basal plate. On that section, you can see the sharp border that goes sort of in the dorsoventral direction between the basal plate and the alar plate of the midbrain. That's the top border, or the bent border there on the top. And on the bottom, you can see that it extends into the diencephalon. It doesn't include all of the diencephalon, so it's actually, this spatial mapping is more precise than our previous annotation. It's really two of the segments, of the anatomical segments of the diencephalon specifically, and not, for example, into the hypothalamus. On the right, you can see the same thing. Now you see all sections, so you can see also how it's distributed in 3D. So you can see it's really located in the medial part of the brain, around, close to the midline. Once you have a projection matrix like this, this matrix that Tangram generates, it's really a tool that you can use to project any property from the single cell universe to the spatial universe. And more precisely, you can impute gene expression. So I said we measured 119 genes, but now that we learned the projection of all the clusters onto space, we can take all the genes that are expressed in all the clusters and project them as well. And in many cases, not all cases, but in many cases, this works incredibly well. Here's an example that I was actually quite amazed by. I was using the gene 6.1 and PRDM12 as markers of cranial ganglia and sensory neurons, because I was trying to locate the cranial ganglia and the DRGs. It's known that 6.1 is a great marker for cranial ganglia, and it's also in the notochord and the DRGs. And PRDM12 has the same expression pattern, but according to literature, it's also expressed in the dorsal midbrain and diencephalon. And indeed, you can see on the left there, if you compare the PRDM12 expression pattern and the 6.1 expression pattern, they are almost identical, except that PRDM12 is clearly detected in the dorsal midbrain and diencephalon, as indicated by the arrowheads. And on the right, you can see also that we can confirm using the Allen Brain Atlas that there is a expression of PRDM12 in both the DRGs and the notochord, and also in the cranial ganglia and in the dorsal midbrain and diencephalon. So this now gives us access to the whole transcriptome in 3D with all these sagittal sections using this imputation method. Now, going back to the organizers that we talked about a few slides back, we can now map them. So now we can say that these clusters that we identified as the ventralizing organizers, they have very specific spatial distributions. So number 208 there is a hindbrain floor plate radial glia. Number 207 is a midbrain floor plate. 206 is midbrain basal plate. 212 is the zona limitans intrathalamica, with this little wedge between the, that goes next to the thalamus. And on the right, you have a floor plate of the hypothalamus. So now we have a much more precise and richer understanding of where these organizers are located. And of course, since these are clusters, we now also know exactly what these organizers secrete and where is the source of these secreted molecules. Same with the boundary organizers. There's a set of organizers around the midbrain-hindbrain boundary, or also called the isthmus, and which has very precise spatial locations, as you can see there. And finally, the dorsalizing organizers at the bottom. Again, there are specific dorsalizing organizers of the hindbrain and the midbrain and the forebrain. And actually, I've been blown away by the unreasonable effectiveness of Tangram on this data set. So we designed our gene set, 119 genes, for the organizers. So the genes were selected from radial glia of the brain, right? But it turns out that we can, using these genes, also map in exquisite detail all kinds of things that are unrelated to the source of this gene set. So here's an example of the eye, where on the left I'm showing the one section where you can see the eye, and we have a cluster number 55, which we identify as the optic cup, or the optic cup and stalk. And in blue we have the lens epithelium, and in green we have a cell type that we identify as part of the early macrophages that I showed before. And you can see on the zoom-ins there, where I show several sagittal sections at the different distances that you see there. And at the bottom of that zoom-in, you see the separate color. So you can see that the macrophages are really located in the middle of the eye, in the same place where the lens is, but they're different cell types. So we're mapping two different cell types to the same place here. And surrounding it, you have the optic cup and stalk that clearly maps to the surrounding region, but not to the sort of pupil area. And we're also able to impute genes that have a known role in eye development. So MITF, VSX2, VAX2, PAX2, and so on. We have measured PAX6 at the top, on the right top. You can see actual expression of PAX6 in the right and left eye. But the ones below are the imputed expression patterns of other genes involved in eye development. So this is just sort of a bonus that we're able to map the eye, and we're doing the same now for the otic vesicle, the ear, and the cranial ganglia, and other parts of development of the head. And it's a very rich data set. All right, so finishing my talk, I wanted to go back here and just make one point of all the things that we learned here. Whoops, sorry. Of all the things that we learned here, I think one thing stands out, and one question stands out, which is what is the origin of the incredible complexity of the brain? Where is the diversity of neurons generated? And I think looking at the organizers and the spatial patterning, you can appreciate there's this enormously complex mechanism to generate diversity among radial glia in early brain development. But still, despite all of that complexity and diversity of radial glia, what you see here is that most of the diversity is generated later. So we found some 50 odd clusters of radial glia generated by all of that patterning and so on. We found probably twice as many neuroblasts and probably 3 times as many neurons in this data set. So since all of this was done in the same way, everything was clustered in the same way, I think that shows very clearly that there is more diversity generated post-mitotically in the neuronal lineage after they left the progenitor state than there is before, despite all the complexity that is already generated at that stage. And with that, I'm just going to mention the companion website, mousebrain.org. We have the adolescent brain atlas there, as well as the developmental. Not yet updated with what I talked about today, but hopefully in a few days it will be. We share all the data, so you can play with it. And I want to thank all the collaborators and people from my group and the funders and you for listening. And that will be all for me.
Thank you, Sten. So again, so you can ask questions in the window below. So I can start. So there's a question from Esther, I think, Serrano. she's on a very nice talk. What about chromosomal sex? Can you plot sex in your clusters like you do with space and time?
Yes, we can. So we don't know the sex of the embryos that we start with because we haven't got a way to see that on the embryos, especially in the early stages. But we can infer the sex of each cell based on the expression of, for example, EXIST, the X inactivation gene. And we've done this. I would say, though, that the sampling was not done in such a way that you can necessarily, for example, look at sexual dimorphism, because we don't have enough individuals in each, time point and tissue. So you would have to sample it, I think, in a different way if you want to look at sexual dimorphism.
Okay. Next question is, even though I missed a similar development map for the spinal cord, so it's from Augusto Escalante. So regarding the spatial mapping of single cell data, would it be possible to discover left-right asymmetries in the distribution of cell types, or is that something we cannot yet achieve with current methods?
No, absolutely. That should be possible with this. There is nothing. We're not imposing any symmetry here. We did do the spatial mapping from one ear to the other, so we have both sides. And the clusters that we observe from single cell data, of course, are, if there is a difference between left and right, there will be two different clusters, and we would map them differently to left and right. It's actually a good suggestion. I haven't looked at that. It would be interesting to have a look.
So the spatial mapping you did and that you show for today, did you also do it at later stages, like 16, 18, etc.? So the data sets will be available, or it's just for the young ages?
We did it for just one stage now.
Okay.
So it's for the E10, the mouse was E10 and a half. And in the analysis, I included all the clusters that have at least half of the cells are from E9, 10, or 11. So it's a window on that segment, that stage of development. Of course, it would be great to have also the earlier and the later, and but you have to end some time and write a paper, and then I'm sure we'll get back to it and try to do more.
Okay, so now a question from Frank Schubert. So very impressive, interesting point about generation of diversity post-methodically. This to some extent goes against the well-established specification of neurons in the ventricular zone or in the spinal cord. Do you think that this implies differences in neuron specification between brain and spinal cord?
I guess potentially yes, but I should say that it's difficult with this data to rule out that there are hidden differences that are established during, for example, the progenitor stage and are only visible later on. So it could be that there is 300 different types of radioglia, just that what makes them different is not visible on the transcriptome level, and that this only becomes visible once they start to differentiate into neurons. So I'm not ruling out that there could be pre-specification necessarily, but I do think it's likely that there is substantial diversity generated, maybe stochastically, later in development, or sorry, later in differentiation of each neuronal lineage.
Okay, thanks. So we have another question from Mikhailo. So do you think chromatin changes could prime neuron heterogeneity at other stages? For instance, more radioglia subtypes possible based on chromatin accessibility, etc.
Yeah, I mean, maybe that's kind of what I was getting at. That's possible, and it's impossible to rule out with this present data, although I think it's also possible that there are... So everything happens quite fast here. Very interesting to see how it looks in human, where everything is much, much slower. There are not many hours between a progenitor might have been specified for something, and then it becomes a neuroblast and an immature neuron. It goes very fast. So it might be that a specification event happens on the radioglia stage, but before we catch this and it's sort of been converted into transcription and it's visible that radioglia specific neuron, a specific cell has already exited the cell cycle and now it looks like a neuroblast. So since everything is so fast here, it's maybe difficult to separate the specification event from the output of the specification event. In human, I suppose that should be more evident. I'm not sure, but that everything is probably 5-10 times slower, so possibly.
Thank you. So now a question from Denis Jabaudon. Thanks, Sten. Could you think of a possibility to model this process? You have access to the transcription diversity of progenitors or of the postmitotic progeny. Is there any meaningful way to try to computationally link the two?
To link the progenitors to the progeny that they generate?
Yeah.
Yes. Yeah, we do that in the paper. We try. It looks like it kind of works. Based simply on having enough, if you have dense enough sampling, you should be able to sort of, like you look in this big Disney map, you know, you can sort of walk from the neuroepithelium and you can walk up to the fibroblast or you can walk to the glioblast. And so you can, of course, systematize that and make it into a computational method. Nevertheless, it's difficult to be sure. So you really would like to have more of a genetic tracer or something to be really sure, like a CRISPR, or there are these new methods now looking at mitochondrial mutations. And this particular data set is not great for that, but I think really to be sure, you need something more physical that really links cells unambiguously.
Thank you. So now myself, I have a question on the organizers that you mentioned. So the special one. So all the different subtypes or subdomain of organizers, they really define each of them by a different, at least one different secreted molecule or morphogen expressed in all of them? Or it could be something else. Or each time you define a subset has a link with a morphogen or something that is secreted.
Yeah. I mean, they were not defined by morphogens. They were defined by clustering. So the differences could be anything. It could be metabolic genes or whatever. But when we then look at the differences between the clusters, we find different secreted molecules or, these kind of molecules. We do find such differences between all of these organizers that are not the canonical ones, not FGF and Sonic Hedgehog and these things, but others that are less studied. So I think the conclusion is that there's potentially a lot of functional ligands being secreted from every corner of the brain, and it's even more complex than we thought.
Yeah. And for the meninges, I also trying to do the same thing, to look at the meninges, whether also there's a spatial distribution of signaling molecules that could influence the brain. But it's known for a few places, but not so many known signaling molecules.
We're making an effort to try to sort out the relationship between the meninges and the pericytes and the perivascular macrophages. Perivascular fibroblasts. It's a bit unclear where the pericytes come from generally in development, and we think we would like to try to sort that out. And we're doing in vitro, so explant culturing of meninges and these kind of things to try to sort this out. It's definitely there are lots of secreted molecules by the meninges, so definitely it could have an effect, but we don't have any particular results yet.
Okay. So another question from Jörg Giton. So regarding developmental cell death in brain regions and across cell types, with your data, can you provide evidence for early determinants of cell death in the brain?
Evidence for what?
Okay. So can you use your data sets to try to map or to find regions where cell death is first initiated in the developing brain?
So I've tried to find a reliable signature for cell death that would be visible on RNA-seq, but I have failed. If you have a suggestion, I think that would be very cool. But I have never seen anyone show cell death in single cell data based on transcriptome only. I think it's a very rapid event, and it may not be reflected in the transcriptome.
Okay.
But I'm open to it. So this will be very cool. I've looked for it and failed. I also asked around for people to, if anybody has any suggestions, but nobody ever came up with anything.
And maybe there's no more questions from the audience, maybe one from me. So regarding the splicing you mentioned, so what is the correlation between the initiation of splicing and some developmental events? So for you, when do you first see evidence of splicing? You mentioned it because a broader view, but so can you link it to something like axon genesis, for instance, or something like that? What is the first?
We haven't looked at any specific classes of molecules that it would, if it would affect a specific class of molecules specifically. And I think that's an interesting question. Now we looked just on the general spliceosome that would affect all splicing equally. And we just see that there is this transition as in roughly the same time point when cells exit cell cycle, they also downregulate the spliceosome, and at the same time, the proportion of unspliced RNA increases. It is interesting to think about. I think it's clear that in neurons there are more splice variants than in other cell types often, and there are many RNA binding proteins and splice factors that are expressed specifically in specific neuronal types. So there might be some functional reason for why you would want to have slow splicing in neurons, but this is just pure speculation.
Okay, so if there's no more questions, so I want again to thank you, Sten, and everybody for listening. And I guess we hope to see you again soon, maybe in Brussels or elsewhere, but probably not before the spring.
Yeah,
thanks again.
Thank you. Thanks everybody for coming.
Okay. Bye-bye, everyone.
Bye.